joining us on today's episode of Myeloma Crowd Radio this afternoon. I'm your host, Jenny Ahlstrom. We'd like to take a minute to thank our episode sponsor, GlaxoSmithKline, for their support of Myeloma Crowd Radio. Now, before we get started today, I'd like to mention that the topic we'll be discussing today with Dr. Keith Stewart has a great deal to do with the genetics of myeloma. And last Saturday, we hosted a Myeloma Crowd virtual roundtable with Dr. Sikander Alawadi, Dr. Faith Davies, and Dr. Brian Van Ness to discuss myeloma genetics. We also have an entire course inside of Heltree University dedicated to myeloma genetics that's taught by myeloma experts, and you can access those resources on the myelomacrowd.org website and on the heltree.org website, because we should all understand our own type of myeloma. So we're excited to have Dr. Keith Stewart with us on the program today. Welcome, Dr. Stewart.
Hi, Jenny. Great to be here.
And let me just introduce you before we start in with questions and the topic that we're here to talk about today. Dr. Keith Stewart is Professor of Medicine at the University of Toronto, Vice President of Cancer at the University Health Network, Medical Director at the Princess Margaret Cancer Centre, Vice President of the Toronto Central South Regional Cancer Program of Ontario Health, and the Richard H. Clark Chair in Cancer Medicine. Prior to this recent appointment, Dr. Stewart was Medical Director of the Clinical Cancer Research Unit at the Mayo Clinic in Arizona, Professor of Medicine and Consultant in Hematology and Oncology at the Mayo Clinic in Scottsdale. He's returning to his roots at the University of Toronto, having started there in 1992. He serves on the Scientific Advisory Board of the MMRF of Myeloma UK and the National Cancer Institute's Cancer Therapy Evaluation Program. Dr. Stewart received the Vasek and Anna Maria Pollock Professor of Cancer Research at the Mayo Clinic for 10 years, has been listed in America's Top Doctors, and was in Canada's Top 40 Under 40 list of physicians in 2002. Dr. Stewart is an editorial board member of the American Society of Hematology, or ASH, Clinical News, and has performed editorial service for blood, current gene therapy, and targeted therapies in oncology. Additionally, he's reviewed articles in over 19 additional publications. Dr. Stewart has research specifically supported by the NCI with a very coveted SPORE grant, as well as other grants on the topic we'll be discussing today, clonal evolution in myeloma and high-throughput drug screening, and correlations with mutational status in myeloma cell lines. He has also performed significant research funded by the NCI on overcoming drug resistance. So welcome again, Dr. Stewart. We're so excited about your work and this topic today.
Well, thanks, Jenny. That guy sounds great. So thank you for the introduction.
So let's talk about this new research that was done while you were at the Mayo Clinic, but I'm sure you're continuing your work there at Princess Margaret. What was the hypothesis to create a new test to screen drugs against different types of myeloma?
Well, Jenny, we had been working on trying to develop our own drug, some work we had done with the Leukemia and Lymphoma Society, and we started partnering with a group of medicinal chemists, or people who make drugs for a living, in California. And at some point in our efforts, one of them, who had had decades of experience making drugs for pharma, said, Keith, the quick way to success in this business isn't what you're doing, it's drug repurposing. And drug repurposing is it's the notion of taking drugs that are used for a different indication and using them to treat cancers or other diseases. The classic example, of course, being thalidomide, which turned into Revlimid, and pomalidomide, which is obviously probably one of history's greatest examples of how this can work properly. Excuse me. So we thought about what they had said and decided that we would try and explore drug repurposing. So we took out of their fridges and freezers that they had not only all the known drugs that worked in myeloma, which in some ways would be a positive control, but also some drugs that we thought might be active but had never really been tested. So that's how we started, and we quickly recognized that there were some opportunities to take the system we'd built and to try and use it to personalize therapy for patients. So, and please interrupt me if I keep going on too much.
Oh no, sure.
What we had built was using robotics. We would take a bone marrow sample from a patient with myeloma. We would sort out from the total bone marrow the tumor cell, the myeloma cells, and then we would place those into tiny little wells with only 1,000 or 2,000 cells per well, and we would use plates with 96 wells on them, or even larger or smaller plates. And then we would use a robot to deliver drugs at different concentrations into the wells to see if they were able to kill off the myeloma cells in that system. And doing that, we realized that this might be a way to personalize care, and we ended up selecting about 80 drugs, which in our studies, both known drugs and some that weren't known to work in myeloma, would be suitable for this kind of testing. So the idea when we started was, let's build now we have these robotics, we know how to do this, it seems to work. Can we build a test that when I take a bone marrow from a patient, within a few days, we can come back to the patient, excuse me.
Oh, you're fine.
Can come back to the patient and tell them that certain drugs would work in their condition and certain drugs would not. So that was the premise of how we started. The important things were we wanted it to be something that would be quite quick to turn around, quite simple to perform, and that would be something that's compatible with getting approval for this to be a clinical test one day.
And did you start this process a long time ago?
Yeah, we probably started maybe, I'm going to guess, about 4 years ago. So it took us a while to establish the system, to do the drug libraries, the library of drugs, to screen that, to see which drugs might be suitable, to refine the conditions. And one of the interesting things we found is that you could tell within 24 hours of adding the drug to the tumor cells that this drug was likely to be effective, and if you waited longer, it would just be more obvious. So that was quite important because for your listeners, myeloma cells, when you take them outside of the human body, die quite quickly, actually. They seem to be very highly dependent on substances in the body that keep them alive. And so they only really survive at best for 3 or 4 days, sometimes 5 days. So we needed something that worked quite quickly and that was reliable.
And I think there's an interesting finding we'll talk about later about some of the IMiDs that they take a little bit longer to work. But
yeah, we'll come back to those because they're a strange breed.
Yeah. You also talked about this being primarily a research tool, but it could end up as a clinical tool. I think patients are just curious about that, and maybe it's just for research, because if you can do high throughput screening on hundreds of drugs, that's a lot faster than guessing, even if it's not a clinical tool.
Right. So our work initially set out to do exactly that, which was to build a clinically relevant test that we might apply to patients. I don't know that we will take it forward as a clinical test for some reasons. Maybe we can discuss later. But it is. There are companies out there that are doing this now, particularly there are some companies doing this in the leukemias. I think there are even 1 or 2 companies, if you search hard enough, that might offer something similar in myeloma. But I think for a variety of reasons, we are going to probably keep this in the research arena for now. You'll probably go into a little bit of detail some of the things we found, but probably at this point it's important to mention that we ended up studying about, in our paper, about 100 myeloma patients, but now through our Biden Moonshot grant for the National Cancer Institute, which is to look at drug resistance, we have studied about 160 patients as well as about 40 myeloma cell lines, which are, of course, immortalized cells that we can use and are replenishable. So with that, the thing that you started off mentioning was genetics of myeloma.
Mm-hmm.
And we decided we would not only look to see what drugs worked in an individual patient, but we would also do genetic studies to see if we could link the responsiveness of a patient to a certain drug to the type of genetics that were driving the tumor to grow in the first place. And that, in some ways, became just as interesting as the concept.
Mm-hmm.
I will tell you that when we sent grants and papers for review, one of the criticisms, which is a valid one, of what we've done is that myeloma cells, as I mentioned already, are quite highly dependent on the natural environment of the bone marrow for survival. And there are many groups, quite rightly, who say if you take that natural survival away, you're probably biasing your drug library screening in favor of killing the cells, because that natural support system, the gas and electricity and water to the house, if you want, it doesn't exist anymore. And so that was true, and we acknowledge that that's a weakness. On the other hand, to create that environment in a way that would be compatible with a clinical test is almost impossible. And so that is why we chose to do something simple and efficient. And we thought that because we could see a result within 24 hours, it sort of fulfilled those criteria.
Mm-hmm. And the speed of death is probably indicative too, right? I mean, that's what you're looking at. And to be able to do such a broad set, I think is just fascinating. You were testing also individual drugs, right? It wasn't like you were doing combinations of drug therapies, or you were just screening a big panel of drugs.
Our first paper we started off with about 300 drugs, and we picked 80. So all the drugs you've heard about and some that you haven't. And so what we do for each patient is we test all 80 drugs one at a time against the patient's cells. And what we did, because sometimes you don't have enough cells to do all 80 drugs, what we did is we put what we thought were the most interesting drugs on the first plate, so that if cells were limiting, at least we could study maybe only the first 14 or I think we could fit 14 drug tests on one plate. So we would go in batches of 14, 28, etc. So for each patient, we'd be able to tell their physician of the 80 drugs we tested. These drugs killed myeloma cells quite effectively. These did not at all, and some of them were in the middle. And then provide that result back to them. Now, because it was research, we couldn't provide it as a clinical test, and they couldn't take action on it. But it was more to try and understand whether it would be helpful or not in a real life situation.
And plus, it might uncover a whole new class of drug, just like the thalidomide accident, essentially.
And it actually has. We think we found a class of drugs that we had not expected to find. And when you talk about, what are we going to do in our research moving forward, you've already hit on the two things we're going to focus on. The first is combinations of drugs. We don't think, and history teaches us, that a single drug is unlikely to be effective against, even in a single patient. There are many flavors of myeloma. And we don't think a single drug is going to capture all of these at the same time. Also, sometimes 1 and 2 equals 4 in chemotherapy worlds, and we wanted to see if we could find drugs that work more effectively together, which we've done in some instances, and I can talk about that later. And then the second part of our research was to explore these new drugs that we found that we think should be effective in myeloma and should be pursued into the clinic in trials.
Okay, now you have me very curious. So I'm making a note that we talk about these new classes, but I'm grateful that you're thinking about all the genetics as well. I've heard other myeloma experts say sometimes that they are surprised at how aggressive what might be considered standard risk myeloma can behave. And then alternatively, some patients have these high-risk features but don't behave as high risk. And I know you have such the Mayo Clinic Scottsdale group was really well known, I think, for the genomics of multiple myeloma. Yeah. Are there any thoughts that you have around that before we jump into some of the findings?
Yes, I think what the audience, the myeloma patients listening, need to understand is the type of genetic testing we do today as a routine clinical test is not particularly sophisticated, and it is only gives sort of a fairly coarse estimate of risk. To get to the full estimate of risk, one needs to do much more sophisticated testing, sequencing of the genome, looking at the RNA, which is the product of DNA, and is what actually is the machinery driving the cell. It's not the blueprint; it's the actual workhorse. And so you can do those tests. Some of them are commercially available. There's a company called Skyline Diagnostics in the Netherlands that offers testing for the RNA, and it's been quite good at showing prognosis, less good at predicting what therapies you should receive. And then when you do genomic sequencing, you actually find that there are many, many more genetic changes present in an individual myeloma patient than we're currently testing for. And, groups like the Myeloma Research Foundation's COMPASS study, which studied over 1,000 patients and continues to be an ongoing study, has found, for example, that you require sometimes you can dissect down to very fine detail what is a very bad myeloma versus an okay myeloma, even though the FISH testing, which most people have done, which is the standard clinical test, indicates low or high risk, and it may not be correct. So one of the shocking findings, actually, some of your audience will have heard about deletion of chromosome 17, or the gene is P53, and that if you have loss of that part of chromosome 17, that you're a high-risk patient. And that's about 10% of all patients at diagnosis, but with each subsequent relapse, it becomes more frequent. And what we learned in the COMPASS study is, in fact, hopefully your audience will know, you have 2 copies of every gene, and it turns out you have to damage both copies, not just one, which was what we're testing for, to be truly high risk. And so part of the implication is a lot of patients that we're telling are high risk are probably not. And on the other hand, there may be some who are standard risk who, as you pointed out, do badly and are clearly are higher risk genetically than we first suspected. It would be naive, however, to say that we understand everything. I think even with genetic testing, there is still a lack of, it's still imprecise. And I think this is why people are beginning to explore things beyond genetics to look at the chemicals and the proteins that ultimately make the clock run in the cell and try and decipher better what's a high-risk patient and what's not, and what makes them drug-resistant and what makes them sensitive, which is sort of why we combine genetics with the drug screening.
Right. And so nowadays, are you suggesting that your patients get either the gene expression profile like the Sky92 test or the next generation sequencing like the Foundation Medicine test when you do genetics?
Well, the Foundation Medicine test would be halfway there. In fact, you can go much deeper than that even. So it's like many things in medicine. The science is ahead of the funding. And so today it's hard to get funding for anything except the basics on a routine basis. But the science is telling us that we should be digging deeper, and that over time that will gradually become a part of our routine workup. But for today, it's usually in the United States, we do FISH testing, and it is usually, in most cases, you're able to get genetic sequencing if you want it done. Myeloma, first the myeloma panel we developed, Dr. Langer at Sloan Kettering, I think, has one. And then, of course, there's Foundation Medicine in Tempest, the sort of commercial labs that will offer this kind of testing as well. So if I were a patient with myeloma, I'd want that done. The question is whether the insurance company will pay for it.
Yeah, and sometimes you kind of have to ask your care team for it specifically.
Yeah, and you can push a bit. And sometimes it can be done on a research protocol, even if it's not a clinically valid test. It can be done on a research basis. So certainly, as you point out, is something our group in Scottsdale and across the Mayo Clinic really focused on for many years, is how to develop these tests and to bring them up as clinically diagnostic. I will say that much of the reason the sequencing hasn't caught on is at this time, much of the information you learn is interesting, but it doesn't terribly alter treatment. The one place where I saw recently that it does alter treatment was some data that the Skyline group had generated, where they suggested that just using their test changed the type of treatment people got almost half the time. So that some patients who were thought to be standard risk became high risk, some high risk patients became standard risk, and their physicians took that information and altered not necessarily the drugs the patient would get, but perhaps how long they stayed on them, whether a second transplant would be used, because this was done in Europe, whether maintenance was with one drug or two drugs. So they made some alteration to therapy based on that test. And I think that will gradually pick up steam. Right now, that test isn't FDA approved. It is available at some centers around the country, and certainly you should be asking your physician if it is available or if similar genetic tests are available at your center.
Yeah, I think especially as you're newly diagnosed or you're relapsing and trying to decide what to do.
I think information is power, so it never hurts to have more information about your disease. And, certainly sometimes it's not what you want to hear, but it's usually better to know that for many reasons. One, it might change your treatment. Two, new drugs might come along that aren't available today, and we'll maybe talk about venetoclax later as an example of that. Three, sometimes it's just better for life planning to know, what side of the fence you sit on.
Yeah, I completely agree. So when you were mentioning the test, you talked about it running it in 24 hours and being within 24 hours, you could kind of tell. Is that kind of the normal turnaround time? Was there a different turnaround time? How long did you let it go also to see? And you mentioned, that myeloma cells are dying outside the bone marrow.
Yeah, we tried 24, 72, and 5 days. And what we saw is if you take 5 days, we lost a lot of cells, so it was a bit noisy. If you looked at 72 hours, it was probably the cleanest time point for analysis. However, we realized that at 24 hours, you could already see that pattern emerging, and not much changed over the next 2 days, except there was more cells dying. So we ended up, because there were more cells alive to study and because it was a faster turnaround, using 24 hours as our metric. Now, you mentioned immune-modulating drugs earlier, Jenny, which are, again, thalidomide, lenalidomide or Revlimid, and pomalidomide or Pomalyst, and iberdomide, which is the new one that's coming. And they all work the same way. Essentially, it's that one after the other, they become stronger. One of the things that I've always puzzled about, and we've studied these drugs for decades now, and which this test really brought home to me, is they are pretty horrible at trying to kill myeloma in outside of the body. They really don't do a very good job. And and one of the reasons our test turned out, I think, not to be as clinically useful as we thought it might be, is you couldn't really tell if the IMiD drugs, these 4 drugs, because we studied them all, were working or not. And even if you waited you could kind of tell at 5 days which ones were responding, which one weren't. And the other thing that people, I don't think have fully explained yet, is you need huge doses of the drugs. I mean, thousands times higher doses of the drugs than you need for other more effective drugs. For example, bortezomib or carfilzomib, the proteasome inhibitors. You need what we call nanomolar concentration, which is tiny amounts of the drug, versus micromolar, so 1,000 times more drug to get the same effect. So why that is true, I have no idea today. I think it suggests that some of the action of those drugs might not be directly related to killing the cells, but more to activating the immune system or causing some arrest of cell growth. Because one of the assay we use measures the inability it measures cell death. It doesn't measure very well if the cell just stops growing. That doesn't help as much. So that's one of the mysteries of life that we haven't figured out yet. Why are these drugs so powerful in humans, and yet if you were to take a lab-based test to try and decide what drugs to use, you would never pick these ones. So, fortunately, through serendipity, they turned out to be very powerful and probably one of the most useful drugs we have in this disease. But had you started where we are and looking for drugs that look good in a test tube, you would never have picked those 3 drugs. So that's a very curious thing, and one that we still don't really understand. And
right,
not
and
to get too scientific about it. Yes,
no, go ahead. Well, it's the name
to...
immunomodulator, right? So they're modulating the immune system, which is maybe why they're only good. I don't know.
Well, I think that, and it may be that they've been very aptly named because of that. They were named that because of the effect they have on some of the proteins that stimulate the immune system. Particularly, they block one of them, and secondly, they increase one of them called interleukin-2. But for many years, I don't think any of us thought that's how they were working until sort of the discovery that CAR T cells were so effective. I think had us re-examine this skill these drugs have to kill cells in humans, that it may actually be due to their immune activation as much as directly killing a myeloma cell. But we're sort of diverging a little bit. These are sort of theoretical things right now. The important thing for patients is they work extremely well.
Well, let's talk about what else you learned, because I was blown away at all the detail that you learned from this study. I was just amazed by it.
Well, it was many years of work with a talented team, and I should give credit to my lab and to Dr. Nathalie Maurice, who did all the drug, and her husband, Joachim Petit, who did all the drug screening, and the staff that analyzed it. So just to summarize for the audience a little bit, some of the things we learned. First of all, the drugs we thought would work really well worked really well. So the proteasome inhibitors, bortezomib and carfilzomib, were consistently at the top of the list of most effective drugs. The immune modulators, which we talked about already, were disappointingly not among that top list. The second thing we learned, there was about 10 drugs that we studied that were completely inactive. Some of the drugs which were, even at the time, in clinical trials for myeloma. So, for example, we studied a drug called ibrutinib. Ibrutinib is approved and used in other leukemias, particularly chronic lymphocytic leukemia, and was in clinical trials in myeloma when we started this study. We saw no activity of that drug at all in myeloma. So, one of the things we point out is if you did this kind of screening, you could probably reject drugs more quickly, or I think more importantly, you could select in the lab patients who are most likely to respond to the drug and enrich your clinical trial for those patients so you would see faster evidence of activity and hopefully get to faster drug approvals. The next thing we found, Jenny, was that by this time the drug venetoclax had sort of been discovered to be very powerful, and I'm sure you've talked about this on your show before, in one particular genetic type of myeloma. And I just in case other people haven't heard this from other places, it's a type of myeloma. It's 20% of myeloma patients, and they have broken chromosome 11, and it's joined to chromosome 14. So a good reason to get genetic testing done today is to find out if you have that type. That particular type turns out to be exquisitely sensitive to the drug venetoclax. Which is approved by the FDA for a number of cancers, not yet myeloma, but approved for a number of acute leukemia. Is really changing how that's treated. Chronic lymphocytic leukemia is changing how that's treated. Anyway, the point of my story is that in our system, we went to look to see, number one, does venetoclax work? The answer is yes, but only 30-40% of the time. And then did it mimic what we're seeing in humans, that it mostly worked in these translocation 11-14 patients, and the answer was yes. So it was a sort of positive validation of the power of the assay, was that this drug was working in the test tube just like it did in humans and in the same genetic subgroup of patients. Conversely, when we looked at selinexor, which, as you know, has recently been approved, we found that rather than being active in good risk patients like translocation 1114, selinexor turned out to be most active in the higher risk patients, those with, for example, loss of chromosome 17 that we discussed earlier. We were also able, Jenny, because we randomly took bone marrows from any patient that was willing to share with us some of their precious tissue, we had patients who were newly diagnosed, and we had patients who were on their first relapse, and we had patients who had many prior therapies. And we were able to then correlate what stage of disease were you at when the drug worked. And again, we found that venetoclax, for example, worked better if you treated people earlier in their disease course. And selinexor, conversely, worked better in people with more advanced disease. And so just as selinexor has been approved for now in sort of more heavily pretreated, higher risk patients, we kind of were able to see that as well in this assay. One of the sort of global questions we asked was, if you just put everything together on one picture and said, where is myeloma most sensitive to these drugs? It was kind of the opposite of what you would expect. So you would think, I've got a new cancer, it should be most sensitive to the most drugs. In fact, we found the opposite. So we found that in newly diagnosed myeloma, less of the drugs were active. And in people who had late-stage myeloma, more of the drugs were active, particularly classes of drugs we don't usually use in myeloma therapy. And the thinking there is that as the myeloma gets progressively more drug resistant, it's growing a bit faster, it has got more genetic mutation, it opens itself up to sensitivity to drugs that don't exist when the myeloma is slow growing and not particularly genetically disrupted. And so that was a sort of surprising finding, but I think maybe explainable. So those were some of the other things we found.
Yeah, it seems like it would be opposite. It'd be much more sensitive. So did you find which drugs were the most sensitive for newly diagnosed myeloma samples?
Yeah, so the most sensitive drugs were the proteasome inhibitors, carfilzomib and bortezomib. There were some other drugs in there, though. Selinexor was also quite sensitive. I mean, it still worked quite well in newly diagnosed patients. It was just more evident in late stages. There's a drug called romidepsin, which is used for T-cell lymphoma that worked very well in our assay, but we don't use in myeloma, so it might be worth exploring some more. And I'm trying to think off the top of my head what was in that top 5 list. And then venetoclax, if you had that particular translocation. Corticosteroids, of course, dexamethasone, more active in newly diagnosed patients, as you would expect. So it kind of, apart with the exception of the immune modulators, it sort of fit the pattern of what we've seen. We did not test daratumumab. We just didn't have that in the panel at the time, and we weren't sure it would work in our assay. So we have never gone back to look at any of the immune therapies like daratumumab or the CAR T or BiTEs that activate T cells, because they wouldn't be likely to work in our assay. I think other drugs like GlaxoSmithKline's drug that's working its way through the FDA.
The antibody drug conjugate?
Probably would work in this assay, but we didn't study it at the time. Thank you. The other drug we found that was very powerful in newly diagnosed patients was the drug panobinostat. And many of you may not have heard of panobinostat, but it is FDA approved for multiple myeloma, particularly when combined with a proteasome inhibitor. And we found it to be extremely potent in this assay. So both panobinostat and selinexor are both approved for treatment of myeloma, both very powerfully active in these assays. The challenge is that they're also somewhat toxic to humans. So they're obviously hitting the right things. They're doing their job, and they were brought forward as drugs because they're doing their job in all these models. But the problem is when you give them to humans, they do other things as well. And so both drugs cause diarrhea and fatigue and low blood platelets. And so for that reason, they haven't perhaps had the either we haven't explored the right dose or combination of those drugs that can be given more safely yet, or because they do hit all the right notes when you look at them in an assay like this, and they should and are probably very powerfully effective if you could give enough of a drug safely to a human.
Mm-hmm. And is that
So
why
that's what we need
panobinostat isn't used as often? Just the side effect profile?
Absolutely, is the main reason it's not used that often. And in the groups of patients it was studied, it was effective but not hugely effective. But if it was less toxic, I think we'd use it a lot because it does work quite well.
Mm-hmm.
And
So you mentioned
Selinexor, I think
no, go ahead.
Yeah, go ahead. No, you go, please, Jen.
Well, I don't want to cut you off from your thought. So you were saying something about selinexor.
Well, I think with selinexor, the company is doing the right things. They're exploring lower doses less frequently in combination with other drugs. And we learned recently when you give a much lower dose with bortezomib, it's very, very effective. And the side effects are more manageable. I actually don't like that term, manageable, but they're a bit less. And so I think it's still a difficult drug for patients to take. But as we explore how to give it more safely, I think there's still opportunity there.
Mm-hmm. And so you kind of went over deletion 17p. What about that selinexor might be better in those patients? Did you find anything else for deletion 17p? And then what about the 1q gain or 414 or the other, or maybe even 1416 that are considered to be more high-risk features?
We didn't have one second, Jim. I'm just going to put you on mute for just 10 seconds, okay? I'm sorry.
Oh, sure.
Entertain the audience.
I might just sing everyone a song. We'll see.
Sorry about that. Somebody
No,
coming to the door of my apartment. So where were we?
We were talking about 1q gain, 414, other deletion 17p ideas or working therapies.
Yeah, I think that's already described the two main findings, which were the 1114 and the deletion 17 patients. We still have a lot of data to go through and analyze because we did look at all 20,000 genes that are in the human genome, and to try and decipher, whether there were patterns of those genes that linked to whether the drug worked or not. The problem you get into a little bit, there's so many different types of myeloma and so many different mutations that despite the fact we looked at 150 patients, it gets down to quite small numbers quite quickly. Mm-hmm. So, for example, there's a mutation of something called IDH, which happens in about 1 to 2% of myeloma patients, and there's drugs you can target that with. But of course, we only had maybe 3 or 4 patients in our whole group, so it's not very much to work on because they're just such a rare thing to see happen. So I think that unless there's something else in the paper I've forgotten about, those were our main findings with respect to genetics, but more work to be done.
Yeah, and then did you see similar things for what you call double hit myeloma? And you might need to explain that, or triple hit myeloma.
Yeah, so double hit myeloma is what I was talking about earlier. Well, it's not really. So we worked with Gareth Morgan and his team in Arkansas, and then they moved to New York. And what we did with Gareth is what Gareth did really, I shouldn't take the credit for this, and his team did, is they looked at large numbers of myeloma patients, maybe 1,500, and then they looked at all the different genetic changes. They looked at the age and the kidney function and the hemoglobin and the performance status of the patient, and tried to dissect out what was the most high-risk patient population. And they discovered there were only sort of 3 things that predicted for very high-risk disease. One of them was double-hit myeloma because there were 2 genetic changes that defined what that would look like. So it could be you have one of the chromosome translocations, so the chromosomes are broken and rejoined, that we know are high risk. You could have one of those, but you had a second change too, which might be deletion of chromosome 17, for example. And we stole the term double hit from the lymphoma doctors because they had already used this to describe lymphoma patients who had 2 genetic changes and were at very high risk for early relapse. And so we came up and just with this term double hit myeloma, and triple hit myeloma means you have 3 bad genetic changes in your cells, and instead of 2 bad hit, which was the double hit. The good news for myeloma patients is only about 6% of all patients studied had double hit myeloma, so 94% of patients did not. So studying that population is going to be tricky in trials because it's quite, it's not that common, which is good.
Yeah, interesting. And then triple hit is just having 3 different genetic mutations.
Yeah, so something like translocation 414 and deletion 17 and amplification of chromosome 1, all of which are individually associated with high risk. If you put all 3 together, that's going to be a difficult case. And the connotation for the physician is you better treat that aggressively or look for new ways to treat it, like CAR-T therapy. And it's not a guarantee that anybody with those changes will do badly, but just on average, they will do less well than other patients, and because everybody still remains a bit unique. So not to despair if you find those things, but rather just look for perhaps alternative therapies or a more aggressive approach to therapy, and you may still do very, very well with those changes. But overall, if you took 100 patients with that and 100 patients without it, the 100 patients with it would overall outdo as well.
Yeah. So let's talk about precursor conditions. Did you have smoldering myeloma samples in addition to the myeloma samples? And then how did what were the difference in responses, I guess, to these drugs?
Yeah, we had some, but not very many, because we tried not to have them because it was a different question. We ended up with about 8 out of the 150, and we just to make this analysis simpler, we just decided to treat those as newly diagnosed myeloma. So I don't have anything impactful or meaningful to tell you about the differences that we found on our studies. I think generally we would expect that smoldering myeloma cells would be slower growing and still quite very sensitive to the drugs we know to work in myeloma, but as I mentioned already, probably less sensitive to the broader panel of drugs which are less specific, I think. So not much to say about that. The treatment of smoldering myeloma is a bit controversial right now, as you know. And I think my opinion is a bit different from others. I don't think treating a myeloma with a single drug like Revlimid or daratumumab, for that matter, is a terribly good idea. Others have shown in studies that your myeloma it takes you longer to develop myeloma if you're on those drugs, which is for sure, and that could be a good thing, just to allow more drugs to become available. I just worry that essentially what you're going to do is breed drug-resistant cells before you need to, and that the best way to get a drug-resistant cell in the lab is to grow a myeloma cell or another cancer cell in low doses of a single drug for a long time. It almost guarantees that you'll get drug resistance. So I just worry a little bit that unless you're really, really clearly going to go on and get myeloma in the next 18 months, I would personally not recommend therapy. And I think if you are going to treat myeloma and you decide it needs treated, then you should treat it properly, and you should give all the same drugs you would give to a newly diagnosed patient. But that's probably a minority opinion these days, and certainly if your physician feels otherwise, you should talk to them about it and go through the pros and cons.
No, that makes a lot of sense. I think if you have it, just treat it, and if you can wait it out and not be on anything, that might be better. You don't make the cells more sophisticated. So I had a question too on cyclophosphamide. So I have I find that a lot of people use cyclophosphamide kind of later on in therapy when things start not working. And I just didn't understand the findings. It sounded like it wasn't helpful, or?
No, it's quite simple, really. So cyclophosphamide is a good drug in myeloma. We used it a lot back in the old days. It's of the same class of drug as Melphalan. They're called alkylating agents. And it's a very good drug. It's also quite easy to take, very few side effects. Can be given orally. In fact, it's probably better orally once a week. And it can play an important role in people's therapy. In fact, as you know, many of your patients will know, we used cyclophosphamide in newly diagnosed patients for many years and still do when sometimes, for example, Revlimid isn't available and the patient's in hospital and you can't get access to Revlimid. There's still a lot of cyclophosphamide used, so very useful drug. Cyclophosphamide is metabolized, so you take what is essentially a prodrug, and it's not active until it's metabolized in the body, and it becomes active. And we forgot this when we set up our screen, and we used the prodrug or the stuff you would swallow, but it had not been metabolized. So, like you, we were a bit surprised at first to see there was no activity, and then we realized, of course, that that's actually a good thing. It proved that our assay was correct because there's no way the drug could work without metabolism. So it was a negative control. That's what we said in the paper. So as expected, the negative control of cyclophosphamide, which has not been metabolized and doesn't work, sort of was kind of useful in some ways to show that the assay wasn't just totally non-specific and just any old poison would kill the cells.
Mm-hmm. And earlier in the show, you kind of hinted that you found these new classes of drugs that were active that have not been tested in myeloma. We can't we can't get to the end of the show and not cover that because that's really important. So, what classes of drugs did you discover?
Well, we've published a paper on this now. So we in our very first screen where we took sort of 300 or 400 drugs and looked at them, and they were sort of drugs that this chemical company had on the shelf. They're called Nanocin. Great scientists there, great colleagues and partners. And I don't know quite why they pulled this one off the shelf. They just think they had it. It was convenient. And the really fascinating thing about this class of drugs is that they were developed by drug companies to treat totally different diseases, essentially to treat inflammatory bowel disease and psoriasis, because they were shown to be able to turn off some of the proteins in the blood, or the genes that are making the proteins in the blood that were driving the inflammation associated with those diseases. So inflammatory bowel disease is an inflammatory condition. There are things called cytokines, which are proteins which usually stimulate cells of the inflammation system to grow faster and to proliferate. And these drugs were developed because they inhibited those cytokines. In our assay, quite amazingly, they were extremely powerful and extremely potent, but not in all the samples, only in about 40% of them, for reasons we don't fully understand yet. Subsequent work on this class of drugs, they're called PYK5 inhibitors, and that's spelled PYKFYEE. There were 3 of them that had all been developed by drug companies that you can sort of buy off the shelf now, and all 3 of them were very powerful, and somewhat surprisingly so, and only in about 40% of patients. And it turns out that these drugs that were inhibitors of inflammation were doing so by blocking this gene called PYK5 kinase, and we went on to publish a paper just last year to show that that result. All 3 of these drugs in this class were very active in about half of the patients, 40% of patients. And that since they'd already been in clinical trials before and were relatively non-toxic in the trials, that these are drugs that were potentially repurposable and could be used in humans. So part of our ongoing work now is to try and make slightly different molecular versions of the drugs and then take those into clinical trials. So that's something we're working on now. We're at sort of the stage, actually, of looking to go to animal studies to see if they're toxic or not in animals, looking at other types of cancers to see if is this just myeloma or other cancers affected. And so far, it seems to be pretty specific to lymphoma and myeloma, and not so much the other cancers. So we think we're onto something here. The drugs that we used are sort of generic chemicals you can buy out of a catalog today. So we think it's important to develop slightly modified versions of those and and take them forward to one day go into clinical trials. So we thought it was quite an exciting discovery, and hopefully will lead to more investigation from other people and maybe a resurgence of use. There is one of the 3 drugs is called apilimod, and there is a company that is studying that in early stage trials in lymphomas. And certainly, hopefully we got their interest. They may want to study their drug in myeloma as well, because it's already in clinical studies. It wasn't the strongest of the 3, but it certainly was the most readily available. And the company, unless they've changed their name, is called LAM, L-E-M Therapeutics. And I haven't spoken to them in 18 months, but I think they're still studying the drug in lymphoma.
Mm-hmm. Well, I think it's fascinating to be able to find a whole new class of drugs that could be working. Are they? Yeah. Yeah.
The way they work is kind of curious, actually. When you treat a cell with them, the cell fills up with what looks like a bunch of, sacks of fluid or balloons. We call it vacuolation. So these big kind of fluid-filled sacks develop in the cell, and then the cell sort of spontaneously dies. And it's not something you see with other drugs when you treat myeloma. So it's something quite unique. So just even understanding how they're doing that and why that's important in a myeloma cell's life is going to be an interesting area to study over the next few years.
I think it's fascinating. I also saw something there about, before we open it up for caller questions, I just had a couple more, that there were hyperdiploid patients. I guess these are patients that have extra copies of chromosomes. But what did you see that worked potentially best for those patients?
Jenny, off the top of my head, I can't even remember.
No, I will have to go back and look too. I'll include it there.
Hyperdiploidy we would consider low-risk disease. So it's about 50% of myeloma patients. It's very odd because it's obviously odd number of chromosomes. It's always chromosomes, 1, 3, 5, 7, which nobody will be able to explain to you why that is. It's very strange. We classify those as lower risk. So some of the again, a little bit more sensitive to the usual drugs and less sensitive to the more potent drugs. So selinexor was a little bit less active in that group, whereas venetoclax, well, not venetoclax actually. I'm trying to think what other drugs might have been more useful. You'll have to remind me because I can't remember.
Oh, well, I'll have to go back. Into my notes.
But I think what people need to know is that class of that type of myeloma is the one, honestly, that has the best prognosis, and therefore it's usually quite exquisitely sensitive to the drugs that we use, and it's one of those types that we feel most confident we're going to get a good outcome for. And fortunately, it's 50% of patients. So these are a little bit more common in older patients than younger ones, and a little bit more common in IgG myeloma as opposed to IgA. And if you didn't know this, the audience, IgA tends to associate with the higher risk genetic changes. Again, we have no idea why. So IgG generally, hyperdiploid disease, a little bit better prognosis. One of the curious facts I often tell people when I'm lecturing to families and patients, the worst bone disease is in hyperdiploid patients often. And the theory there is that, so paradoxically, even if your X's look terrible, it may not be such a bad thing in the long run. And the reason for that is we think this disease just is around for longer and it has more time to do damage before it gets picked up because it's more slowly growing and it's causing less problems initially until the bones get quite damaged and then you start to notice it.
Yeah, I've heard that too. Higher bone damage, but lower risk.
Yeah.
Interesting.
Exactly.
So my last question, I guess, would be, is this a methodology that you can use to work with pharmaceutical companies to rapidly find new opportunities? I mean, I would think that you could do really rapid drug screening and save them a lot of time and money and effort to see if something like this would even be workable in myeloma.
I think that's exactly probably the biggest take-home message from this. We even like for venetoclax, for example, rather than doing a trial on 100 patients to show that it works 20% of the time, which is what the data end up showing. If you took that same drug and made sure it worked first, you could probably double the response rate or triple it and have a much faster readout that your drug is effective. It may not work for all drugs. We already talked about the immune modulators and how that didn't work, and the cyclophosphamide, because it wasn't metabolized, didn't work. But for things like bortezomib, and then some drugs like bortezomib work almost 100% of patients, so it wouldn't really help there except to tell you you've got a good drug. But for drugs with that sort of more targeted nature, like the new ones we discovered, or like venetoclax, it could be a very powerful tool.
Yeah, I agree. I mean, whatever we can do to save time and money and get to answers faster about what potentially could cure myeloma, that would be terrific for everyone.
Exactly.
Amazing. Well, Dr. Stewart, I want to open it up for caller questions as well. So if you have a question for Dr. Stewart, you can call 347-637-2631 and then press 1 on your keypad. And we'll start with caller 264-7609. Go ahead with your question.
Hi, Jenny. It's Jack Aiello. And Dr. Stewart, it's always good hearing from you.
Hi, Jack.
I had a question on the assay. You know, we myeloma patients always go to presentations, and one of the first graphs we see is that the deeper the response you get, the longer progression-free survival and overall survival one typically has. Are you able to measure kind of depth of response in this assay?
No, Jack, we can't measure that. All we're really measuring is we end up measuring something called the no, not the way you would think about it. The assay tells you two things. It tells you what percentage of the tumor cells die.
Right.
So it's 100% die, or sometimes it was quite interesting. We saw that 20 or 30% of the cells would die, but the other 70% would just quite happily keep living, suggesting that there was, two different flavors of tumor in the sample, or myeloma in the sample. The second thing it tells you is what concentration of drug is required to do the killing. And so we use something called the effective concentration 50%. So what we would read out to the physician is, this is the concentration of drug it took to kill 50% of the cells in 24 hours. So that doesn't really tell you much more than that. It doesn't tell you whether, in an individual patient, that drug's going to get rid of 99% or 10%. And part of the reason is also, we all metabolize drugs differently. So you give the same drug to two different people, different sizes, different weights, different genetic makeup, and it's not always going to be better liver function, worse kidney function. You're going to get different concentrations of the drug, and it will do different things. So that's why we can't really tell what the depth of response will be. We're just trying to predict whether there's going to be some response or no response.
Thank you, and enjoy the Maple Leafs.
Oh, thanks. Well, that would be a long time coming. But it's sad as a Canadian. I don't even like hockey that much, but there you go.
All right, thanks
for
the question,
tell them.
Jack. Okay, we have another call at 373-9626. Go ahead with your question.
Yeah, Dr. Stewart, it's your favorite patient, Chad Snow. I don't know if you remember me from Phoenix. But
hi, Chad. How are you?
I'm doing good. Hey, two questions. One, you and Jenny had talked at the beginning of the show about getting more accurate genetic testing. Is that something that you can just ask your doctor to order for you, or how do you go about getting that?
Yes, I think you should discuss it with your doctor and say, you've heard it might be useful and that you'd like to pursue that if you can. I think there'll be a couple of questions that might come up. If you're willing to pay something out of pocket, whether you want to join a research study, or just to find out what your insurance will cover in the first place. So I think we're very variable depending on where you are. I think if you're in the major centers, you're going to be able to get that testing quite easily. If you're in a smaller community setting, it may require a referral to a major center to get that kind of testing done. And Chad, by the way, you need to call me afterwards because I know you had some issues recently. I'm glad to hear your voice actually. So give me a shout.
I will for sure. Second question, just real quick. As you know, I was supposed to go into a CAR T trial in March, and that got derailed because of COVID. How is it looking for FDA approval of just commercially available CAR T?
Yeah, great question. So the one that was filed, the only one that's been filed with the FDA so far is from Bristol Myers Squibb, which used to be Celgene, of course, and it's a BCMA targeted CAR T. My understanding is when they filed initially, there were some issues with the manufacturing portfolio they submitted, and they had to go back and fix that and resubmit it again. And the last I heard, and it may not be completely accurate, so don't hold me to it, was they have refiled, and it's in the works. So we used to tell all our patients we expect to see this, and personally I think there's absolutely no doubt it will get approved. We expected to see approval this summer, I think, because of that manufacturing delay and perhaps because of COVID slowing everything down. It may be into the fall before we see a commercial product. We're all waiting anxiously because there's huge pent up demand for this, and now it's not only not commercially available, but the clinical trials also got completely disrupted by the COVID pandemic. Manufacturing problems, but inability to manufacture during the pandemic slowed down availability. The drug companies couldn't come in and study the charts of patients in the hospital, so a lot of the trials were just halted and stopped, and it's been a bit of a difficult situation for everybody. But for commercial product, I think. By the way, I want to just tell your audience that FDA approval doesn't automatically mean the drug's on your doorstep the next day. Once they approve it, then there is a period of time during which the insurers have to catch up. The supply chain has to kick into action to deliver across many countries. And the experience in lymphoma is to some degree that even the pharma companies themselves are having a little bit of trouble manufacturing enough of the product to satisfy the demand. So there are lots of reasons that this might be a bit slower to roll out than everybody had hoped. In the meantime, there are a lot of clinical trials going on, if not with CAR T, with things called BiTEs, and they seem to have pretty active as well. And if you can't get hold of CAR T, I would suggest you talk to your doctor about whether a BiTE therapy on a clinical trial might be of interest. Unfortunately, those are still in pretty early stage testing, which means there's again more demand than supply. So you have to persevere, I think, to try and get on one of those studies. But they do seem to work quite well, and in some patients for quite a long period of time.
Okay.
All right. Thank you.
Thanks for questions, Chad. Thank you, Dr. Stewart, for answering. We have one more question. 558-8163. Go ahead with your question.
Hi, Dr. Stewart. My question is, how do you use this test panel to test future immunotherapies, and where do you go from here? Do you test immunotherapies now, or more drugs in different classes, or anything else?
Yeah, I didn't know if I heard all that correctly, but we haven't really used this assay to test immunotherapies because there is no immune system in the wells. The T cells that are activated by CAR T therapy are not present, so we wouldn't expect to see an activity. So we haven't really. The assay doesn't lend itself to studying the immune system. I think there are other groups out there trying to do that, where they are trying to study that in the test tube, but it's still a ways out from being clinically impactful. Small molecule wise and new drugs coming along, nothing I've really heard about that to get excited about. We certainly are looking forward to getting venetoclax approved by the FDA. We think that the CAR T and the BiTEs will get approved over time, so there's more coming. But new classes completely out of the blue and very active. There may be some. If there are, I'm not aware of them. I mean, the good news, I think, is every 2 years we get a new drug in myeloma, and that's been consistent since 2000. So 20 years on, we have 10 drugs now, and there's no sign of that slowing down. There's dozens of drugs in the clinic, and there's at least 4 or 5 that should get FDA approval in the next few years. So they keep coming.
Yeah, it's amazing. Okay, thank you so much for your question. And Dr. Stewart, thank you so much for joining us today. What an informative body of research you've been working on. I think it's truly moving the field forward in knowing how to treat which kind of patient, how to personalize medicine. And I just say keep going with this assay. And keep testing new things, and we're excited for your research and excited that you're at Princess Margaret, and just appreciate everything you do for patients.
All right. Take care, everybody. Thank you. Bye-bye.
Okay, thank you so much, and thank you for listening to another episode of Myeloma Crowd Radio. We invite you to join us next time to learn what myeloma research means to you.
Okay, round two. Name something that's not boring.
A laundry? Ooh, a book club. Computer solitaire, huh?
Ah, sorry, we were looking for Chumba Casino. That's right, ChumbaCasino.com has over 100 casino-style games. Join today and play for free for your chance to redeem some serious prizes. ChumbaCasino.com. No purchase necessary. Void where prohibited by law. 18+. Terms and conditions apply. See website for details.